Thank you, Kieran. Well, the importance of food safety to the consumer is obvious to everyone, and indeed the obligation to provide assurance of food safety by the industry is also clear. But what is the role of the scientist in all this? The role of the scientist is to provide improved detection techniques, whether that is for a microbial contaminant or a chemical contaminant. We want to improve these methods. However, that is not without its challenges. These contaminants, again, whether they be chemical contaminants or microbial contaminants, are often present in minute quantities. What we're essentially looking for is that needle in the haystack. But it's very, very important that we find that needle. When you consider that just less than 10 bugs are enough to make you very, very ill and indeed kill you. Therefore, we have to be able to find them, and we have to be able to find these contaminants fast. Traditionally, the methods that we have used have been quite laborious and often take a number of days to prove that the contaminant is there, and this is something that we want to address. EU traceability projects have resulted in better detection methods, and these methods can help both industry and the consumer by providing better assurance. Asking the question, have we made the needle bigger or have we made the haystack smaller? I would suggest that we have actually done neither. What we have done is we have improved our methods of finding that needle within the haystack. I'm now going to take a whistle-stop tour through some of the detection methods which have been developed through these traceability projects. Our aims have been to improve detection in a number of areas. We've wanted to improve speed, take detection time from days down to hours. We want to improve sensitivity. Being able before to detect maybe parts per million, we now detect parts per billion. We want to make these methods less laborious, more cost effective, more user friendly and easily implementable. By doing this, we can therefore improve safety, and we can decrease pathogen movement or even chemical contaminant movement through the food chain by being able to spot it faster. Also, many of these tools can be easily integrated into supply chain management, and you will see from the exhibitions how this is being done. First of all, I'm going to look at a whole suite of molecular assays which have been developed. As Jan mentioned in the previous presentation, DNA provides us with a fingerprint, indeed for each one of us, but also for every single microorganism out there. It has a unique DNA fingerprint. And we have exploited this to develop amplification technologies for common foodborne pathogens such as Salmonella, E. coli, Campylobacter, Clostridium botulinum, or bioterrorism threats such as Bacillus anthracis. We now have assays which can be used to sensitively and specifically detect each of these and many, many other organisms. Importantly, these are one-day tests. Results within the day. This allows for rapid product release. The results can be implemented into supply chain management. And indeed, some of these assays are currently in use in the food industry. For example, the Salmonella assay is currently being used by the Danish pork industry. We can also use molecular assays for typing and tracing foodborne pathogens, because not all pathogens are the same. We can use these sort of DNA fingerprints to monitor the pathogen's movement through a process. This allows us to identify key areas of concern that need to be addressed. Or it can also address the question of how virulent this pathogen is, because not all pathogens are of equal virulence, and that is something we need to consider as well. And this is just one example of an assay which has been developed for Campylobacter, which can be used to track its movement and also assess its virulence. Toxins are also common contaminants of food products, and we have also looked through a number of projects at the development of improved toxin detection techniques. Microfluidic devices have been developed for the detection of Staphylococcus enterotoxins, which is a step towards the development of high-throughput, semi-automated microdevices with detection limits in the nanogram range, and these result in reduced time and costs, as we have no processing or no extraction needed prior to doing the assay. Aflatoxin detection has benefited from immunological assays which have been developed. These assays can now be done in 35 minutes, under an hour you have your result, with a detection limit in the picogram range. So this is very, very beneficial for industry. Fecal matter from animal fecal matter has been identified as a key source of pathogen contamination of animal carcasses and therefore onto subsequent meat products. So this is something that we wanted to address, and we wanted to improve the detection of fecal contamination on carcasses. The approach that was taken was, first of all, the identification of a suitable fluorescent marker. This is then added to the animal feed before slaughter. And then the fluorescent marker is excreted in the faeces, which can then be detected by spectroscopic imaging. And here in these images, on the left-hand side, you can see what the eye sees. So you can see the faeces on the piece of meat, and on the right-hand side, you can see the spectral imaging, and you see quite a similar story. However, when we remove the visual faeces, on the left-hand side you can see that the faeces is essentially no longer there, or so you think. However, when you look at the spectral imaging, the faeces is still visible, and therefore is still a potential carrier of pathogenic organisms. After a water rinse, the faeces is completely invisible to the naked eye. However, with spectral imaging, we can still see some traces. And what's very nice about this technique is that it can be implemented online in the abattoir, and indeed has been done so. Looking at the chemical side, we've also developed a number of different methods for chemical contaminants through these traceability projects. This is just one example of a HPLC MS method which has been developed for the simultaneous detection of 38 antihelminthics in bovine liver. The simultaneous detection results in increased sample throughput, reduced waste, improved efficiency, reduced costs. This method has been fully validated and has been made available to the industry for use. It has also been transferred to the national reference laboratories across Europe, and also through numerous training sessions the method has been transferred to INCO countries. The illegal use of growth promotion agents has also been addressed. Biomarkers have been identified which are